fast. Is it going? Do we know if it's going? Start video.
Here we go.
Oh, there we are.
Go
You
ahead.
can see my forehead. Okay, so we're going to be doing 1 through 5 of lesson 11 group problems. My name is Jacob Underwood, and I am the one assigned for numbers 1 and 2. So for 1A, indicate the group of enzymes that covalently link amino acids to tRNA molecules, and that would be aminoacyl tRNA synthases. Indicate 4 proteins that consume GTP while during translation, and that would be EF-TU, IF2, IF3, and EF-G. 1C, indicate 1 molecule that functions as a ribozyme during translation, and that would be peptidyltransferase. D, indicate 2 proteins that recognize stop codons, RF1 and RF2 both do that. Indicate 1 molecule that forms hydrogen bonds with the SD sequence in mRNA, that would be 16S RNA. And the next ones are fill in the blanks for words. The first amino acid in prokaryotic proteins is methionine. An E. coli protein called F2 or EF2 delivers charged tRNAs to the A site of the ribosome during translation elongation. The initiator tRNA binds to the P site of the ribosome, whereas the remaining charged tRNAs bind to the A site. EF-G is a translation factor that helps the ribosome move from 1 codon in the 3 direction along the mRNA. IF3 is a translation factor that guides the mRNA to 30S ribosome subunit. The AUG codon is the start codon, while the UAG is an example of 1 stop codon. There's a lot of them, but really just 1 start codon. And degenerate is a term that indicates that more than 1 codon can specify the same amino acid, or for someone stupid. And the final amino acid is located at the C terminus of the polypeptide. Now we will have Joshua, and he'll be explaining number 3 for us.
All right. Thank you. All right, so number 3 says a series of mRNA codons are indicated below. Consider the wobble rules and provide all possible tRNA anticodon sequences. All right, so we have UUG for the mRNA, and then these are anti-parallel, so you'd have a 3' to 5' for the tRNA, and you would have possibilities, because this is the wobble nucleotide, and you have possibilities of C, U, or A, and for the others, you would have possibilities U, A, G, and I. And then for C, you would have possibilities G, A, and I. For U, G, A, U, and I. And you see the pattern going on. And U and I for A. So these are the possible combinations that you could have for this amino acid to have this tRNA anticodon. And now I'm going to turn the time over to Nick.
4 and 5. Okay. So this one is kind of talking about a little bit more math related to proteins and how to calculate different values. So the first part is we have a protein X, which is 218,000 kilodaltons in size. And then it says that the average amino acid is only 125. So if you divide those two numbers for part A, you can find out how many amino acids there are, which is 1,744. The next one is asking how many nucleotides long that mRNA coding region is, and we know that there's 3 nucleotides per codon, and so you just take the number of amino acids, multiply by 3, and you have 5,232 bases in length for that mRNA coding region. And then C is asking how many GTP molecules are consumed to synthesize each copy of protein X. And for that one, so there's 2 GTP that are required in the initiation stage. And then there's 1,743 peptide bonds that are formed. And that's because we have 1,744 amino acids. So it's like when you separate a piece of paper into 4 different vertical sections, you have to draw 3 lines. So there's 1 less than the amount you need. And so that's why it's 1,743. And then there's 2 that are required to make each peptide bonds. And then there's also, and I just realized I forgot to write that, but then 1 required. So imagine there's a +1 there for termination. And that makes 3,489 GTP molecules required for making protein X. And then ATP molecules required is 1,744. And that is because per amino acid in the chain, you have to have 1 molecule of ATP come into whichever amino acyl tRNA synthase required to make that amino acid to charge the tRNA. And so you have the ATP go in, and then an AMP and 2 inorganic phosphates pop off. So you just have 1 ATP per amino acid. And then last part is, what is the name of group enzymes that consumes ATP? And I already mentioned that, but that is amino acyl tRNA synthetases. And then for question 5, that's just asking you which proteins or nucleic acid sequences are involved in translation. So for A, it's asking which of the above are involved in translation initiation, and that's IF2, IF3, the Shine-Delgarno sequence, the tRNA FMET, and the 5' AUG to 3', which is the start codon. And then for what is involved in elongation, there's EF2, EFG, 16S rRNA, 23S rRNA, and the lysyl tRNA synthase. And the lysyl tRNA synthase is a synthase that is under the amino acyl, and the lysyl is just for the specific amino acid lysine. And then termination, we have RF1, which is release factor 1, and then UGA, which is a stop codon. And then it's asking what are the components of the 70S ribosome, and that is the 16S rRNA and the 23S rRNA. And then which of the above is found within the mRNA. And so you just have your stop codon, your start codon, and your Shine-Delgarno sequence, which is the clamp site for the ribosome. And that is it for this week's problem.
Good job, Nathan.